You must have read about the cellular and structural organization of animals. They have peculiar organs that perform particular functions, and these organs are coordinated synchronously to make the animal fully functional and healthy. But what about plants? Have you ever thought whether they have organized tissues and organs? Well, let us find out. Flowering plants are of two types. They are dicotyledonous and monocotyledonous. Dicotyledonous plants contain two cotyledons in their seed, whereas monocotyledonous plant contains only one cotyledon. These plants also differ in their anatomy, that is, their tissue organizations. Let us discuss the structural organization of the roots and stem. First, let's discuss the dicot root. The structural organization in dicot roots is as follows. The outer layer is called the epiblema, which may protrude out in the form of root hairs. Then the subsequent layer is the cortex, which is made up of parenchymatous cells. Now, can you guess what is the innermost layer called? It is made up of a single layer of barrel-shaped cells without any intercellular spaces. The endodermal cell have water impermeable and waxy substance called Casparian strips. Enclosed inside the endodermal layer is pericycle and pith. The pericycle lies just below the endodermis towards the center and is many cell thick. Can you guess the function of pericycle? They are meant for lateral root growth and vascular cambium growth. The pith lies in the middle. A dicot root usually has 2 to 4 xylem and phloem patches, and later the cambium ring develops in between the xylem and phloem. As you can see in this image, the cambium ring has developed between xylem and phloem. The inner side of the endodermis, the pericycle, vascular bundle and the pith constitute the stele. Well, monocotyledonous root have similar structure as dicotyledonous root. It has root hairs, epidermis, cortex, endodermis, pericycle and vascular bundles. But there are differences in the xylem organization. Monocotyledonous have a greater number of xylem bundles as compared to dicotyledonous roots. Monocotyledonous roots do not undergo secondary growth since they lack vascular cambium in between the xylem and phloem. Now, let us study about the dicot stem. A typical dicot stem consists of epidermis, cortex, endodermis, pericycle, vascular bundles and pith. As you can see in this image, the cortex is seen just below the epidermis layer and vascular bundles are arranged in a circular ring where phloem is seen towards the periphery and xylem is seen towards the center. In between the phloem and xylem, we can find a ring which is cambium, and towards the center of the stem we find the pith. Can you now guess what is the protective tissue covering the plant called as? Yes, it is called the epidermis, which is basically present throughout the outer surface of the plant as a protective covering. Since the epidermis functions as a protective covering, then don't you think it must be resistant to water as well? Epidermis is covered by cuticle which is water resistant. Epidermis bears trichomes which can be in the form of any outgrowths such as hairs, scales, etc. They function as trap for insects, and some studies have shown that they play a key role in the plant's development. We will now move on to the cortex and pericycle. The region between the epidermis and the pericycle is called the cortex, which consists of three subzones: outer hypodermis and collenchyma, middle parenchyma, and inner endodermis. The outer hypodermis and collenchymatous cells helps to provide mechanical strength to the young stems. The middle layer is made up of parenchymatous tissues, and they are thin walled cells. The inner layer, which is made up of endodermis, contains starch sheet since it is rich in starch. The pericycle lies near the vascular system. The medullary rays are present in between the vascular bundles and are made up of parenchymatous cells. Keeping aside all the tissues discussed, do you know how are the vascular bundle arranged in the dicot stem? Well, the vascular bundles are arranged in a ring-like manner. Each bundle is conjoined, open, and has end-dark protoxylem. End-dark is the arrangement in which the first form primary xylem, that is the protoxylem, is arranged towards the center, and the later form xylem, that is the metaxylems, are arranged at the periphery. The center of the stem is called the pith, which is occupied by rounded parenchymatous cell with large intercellular spaces. The last structure that we are going to discuss is the monocotyledonous stem. The monocotyledonous stem is different in tissue organization, wherein the hypodermis is made up of sclerenchymatous cells. Vascular bundles are scattered, conjoined, and closed. Each vascular bundle is surrounded by sclerenchyma and a large parenchymatal ground tissue. Phloem parenchyma is absent, while water containing cavities are present within the vascular bundles. As you can see here, in this image, you can see that the vascular bundles are numerous and are scattered all over the ground tissue. Here, the pith is absent. This image shows the cross-section view of monocot stem under the microscope. Nature is mind-blowing. It never fails to surprise us. Who would have thought that structural organization of stems and root would differ in monocots and dicots? It does not end here. Do you want to find out the structural difference in leaf? Watch on to learn the complete details and difference in organization of monocot and dicot leaves in our next video. Let's now summarize what we have learned.
Manorama Horizon. Learning made simple.